students, and a warm welcome. Hope you all are doing fine. Myself, Dr. Tripti Malik Ahuja. In this video, you will get the solution of the question paper of MSc Applied Microbiology entrance exam conducted by BHU in the year 2018. In the first part, you will get the explanation of the first 60 questions. This is the second part. The link for the first part is given in the description box below and in the I button here. There are a number of videos which will help the students who are preparing for the CUET PG entrance exam. The link is given in the description box below. These videos will help the students who are preparing for the entrance exam for admission to the courses MSc in Microbiology, Biotechnology, Biochemistry, Lifesciences, Bioinformatics, and Forensic Sciences. In the description box, you will also find the links of some books which you can purchase from Amazon. These books will help in the preparation of the general part, which comprises of first 25 questions of the CUET PG exam. There are also some other books given there which are based on the multiple choice questions from different subjects of life sciences. So now let's get started. Before I start, I request you to take out a paper and pen. And as I am reading out the questions and the options, then you can write the option which you think is correct. You can match your answer with that of mine. If it is correct, then it's well and good. If it is not correct, I will also explain the correct answer. Still, if you're having a doubt, you can write it in the comment section. I will give reply to each and every comment. So question number 61. How many carbon dioxide molecules exit from citric acid cycle? The options are 1, 2, 3 and 4. The correct option is the 2. 2 carbon dioxide molecules are produced from each acetyl coenzyme A produced in the citric acid cycle. The question number 62. Listen to it carefully. Which one of the following immunoglobulins is associated with anaphylactic delayed hypersensitivity reaction? The options are IgE, IgA, IgG and IgM. Now, there is an error in this question. The question says anaphylactic delayed hypersensitivity. But actually, anaphylactic hypersensitivity is not delayed, it is immediate. And if it is a delayed hypersensitivity, which is the type 4 hypersensitivity, then that does not involve the Ig molecule, it involves the TDTH cells. So here the question should be associated with anaphylactic immediate hypersensitivity reaction. So according to that, the correct answer is the IgE. Question number 63. A population of individuals or species having genetic differences is referred as the option ecotype, number 2 ecad, number 3 ecotone, number 4 biotype. So I hope you have guessed the correct answer, and the correct one is the option 1 ecotype. Another simple question, the question number 64. Which one of the following gases is microbiocity in nature? Option 1 nitrogen, option 2 ethylene oxide, option 3 hydrogen, option 4 oxygen. Out of this, the correct one is option 2 ethylene oxide. Question number 65. Edmonds reagent is preferred for sequence determination of a protein because during one cycle of reaction it number 1 modifies and cleaves only N-terminal amino acid residue. The option 2 says modifies and cleaves only C-terminal amino acid residue. Option 3 cleaves N-terminal amino acid residue in native form. Option 4 cleaves C-terminal amino acid residue in the native form. So here the correct option is the option number 1. Edmond reaction is a method for the sequencing of amino acids in a protein molecule. In this reaction, phenyl isothiocyanate PITC sequentially degrades the amino acid at the N-terminal residue. Question number 66. In a dipeptide, peptide bond is generated between number 1 option is alpha carboxyl of first and alpha amino of second amino acid. Option 2 alpha amino of the first and alpha carboxy of second amino acid. Option 3 beta oblique gamma amino of first and beta oblique gamma carboxyl of second amino acid. Option 4 alpha carbon of first and alpha carbon of second amino acid. The correct option is the option 1 alpha carboxyl of first and alpha amino of second amino acid. Question number 67. This question is also from protein. So proteins, they make a favorite topic for the question paper setting. Listen carefully. Which one of the following stabilizes alpha helix structure of a protein? The option 1 peptide bond, option 2 disulfide bond, option 3 ionic bond, option 4 is hydrogen bond. So the correct option is the option number 4 hydrogen bond. Alpha helix structure is stabilized by the hydrogen bonds between an amide, hydrogen of one amino acid and carbonyl oxygen 4 amino acids away. Question number 68, also from protein. Exposure of a native protein to heat results into partial denaturation of the protein due to breaking of 1 disulfide bonds, 2 hydrophobic interaction, 3 hydrogen bond, 4 peptide bond. The correct answer is the option 3 hydrogen bonds. Question number 69. Enzymes which do not follow normal Michaelis-Menten kinetics and exhibit cooperativity are 1 isoenzymes, 2 coenzymes, 3 allosteric enzymes, 4 enzymes. The correct answer is the option 3 allosteric enzymes. The enzymes of allosteric enzymes, the examples are glycogen phosphorylase and phosphofructokinase. Question number 70. Lactate dehydrogenase belongs to which major class of enzymes? Option 1 ligase, option 2 transferase, option 3 oxidoreductase, option 4 is isomerase. The correct answer is the option number 3 oxidoreductases. Oxidoreductases are the enzymes which catalyzes the transfer of electrons from one molecule. The molecule is called donor to the another molecule which is called the electron acceptor. Question number 71. Identify an aldose from the options given below. The option 1 is dihydroxyacetone, Option 2, glyceraldehyde. Option 3, xylulose. Option 4 is ribulose. So out of these 4 sugar, one is the aldose sugar, other are the keto sugar, and the correct answer is the option 2, glyceraldehyde. Question number 72. Which one of the following pairs represent an isomer to each other? Number 1, D-glucose, L-glucose. Number 2, alpha D-glucose and beta D-glucose. Number 3, D-glucose, D-mannose. Number 4, D-glucose and D-fructose. So the correct is the option number 4, D-glucose and D-fructose. These are the structural isomers that differ with respect to their functional group. Glucose has an aldehyde, while the fructose has a keto group called, so these isomers, they are called the aldo-keto isomers. Others are the optical isomers and enantiomers. Question number 73. Identify the glycolytic enzyme which is associated with substrate level ATP synthesis. 1, phosphofructokinase. 2, hexokinase. 3, pyruvate kinase, and the 4 is the aldolase. The correct one is the phosphofructokinase. It is also the rate limiting enzyme, and there is the formation of ATP from ADP and a phosphorylated intermediate. The question number 74. Identify a decarboxylase out of the TCA cycle enzymes given below. Option 1, isocitrate dehydrogenase. Number 2, succinate dehydrogenase. Number 3, fumarase. Number 4 is the malate dehydrogenase. So here the correct answer is the isocitrate dehydrogenase. It causes the conversion of the alpha ketoglutarate to the succinyl coenzyme A, and a carbon dioxide molecule is produced. Question number 75. The F0 domain of the mitochondrial F0-F1 complex is named so because it represents: number 1, the protein fragment given no number. Number 2, the protein fragment that does not perform catalytic function. Number 3, cofactor binding domain. Number 4, domain that confers oligomycin sensitivity to the complex. Here the correct option is the option number 4, domain that confers oligomycin sensitivity to the complex. Actually, it is not F0, it is not F0, but it should be FO. But it is a misnomer, so it is written as the F0. The O stands for the oligomycin, which is a natural antibiotic that is able to inhibit the ATP synthesis. Question number 76. Which one of the following enzymes synthesize the cell signaling factor? Option 1, cyclooxygenase. Option 2 is cytochrome oxidase. Option 3 is cytochrome Q reductase. Option 4 is coenzyme A reductase. So the correct is the option number 1, cyclooxygenase. Cyclooxygenase is also known as prostaglandin antoperoxide synthase. It gives rise to prostanoids including thromboxane, prostaglandins from the precursor molecule arachidonic acid. Question number 77. Again a very simple question. Hope you will judge the correct answer. The protein part of an enzyme which utilizes cofactor for its catalytic function is known as 1, apoenzyme, 2, coenzyme, 3, holoenzyme, 4, native enzyme. So here the correct one is the option number 1, which is the apoenzyme. The apoenzyme plus cofactor together it forms the holoenzyme. Question number 78. Which one of the following is an amphoteric molecule. Number 1, alpha glycine. Number 2, triglyceraldehyde. Number 3, sucrose. Number 4, phospholipid. And the correct answer is the option number 1, alpha glycine. Alpha glycine is an amino acid, and amino acids, these are amphoteric because they can be either negatively charged or the positively charged depending upon the pH of the solution. Question number 79. Out of the following lipids, which contains the maximum number of fatty acids? The option 1, cholesterol. 2, biological wax. 3, prostaglandin. Number 4 is triglyceride. So here the correct option is the biological wax because they can contain the the fatty acids which can be more than 30 also. While cholesterol, it usually contains 27 carbons in the fatty acid chain. The prostaglandins, they contain 20, triglycerides, they contain 3. Question number 80. In a nucleotide structure, phosphate is attached to the ribose sugar by a number 1, phosphoester bond. Number 2, phosphodiester bond. Number 3, glycoside. Number 4 is peptide. So this is a very simple question, and I know you know the correct answer, and correct answer is option 2, phosphodiester bond. Question number 81. 2'-deoxycytidine is a 1 nucleotide. 2, dinucleotide. 3, modified base. 4, nucleoside. So again it is very simple, and the correct answer is the option 4, which is nucleoside. Question number 82. Which one of the following RNAs assume tertiary structure for its function? Option 1, hnRNA. Number 2, messenger RNA. Number 3 is the tRNA. Number 4 is the 5S ribosomal RNA. So the correct answer is the option 3, transfer RNA. Transfer RNA has the clover leaf structure having the D and T loops. Question number 83. During prokaryotic DNA synthesis, RNA primers at lagging strand are removed by option 1, S1 nuclease. Option 2, DNA polymerase 1. Option 3, DNA polymerase 3. Option 4 is RNase 2. So out of this, the correct option is the enzyme DNA polymerase 1. The option number 2 is correct. The DNA polymerase 1 removes the RNA primers by exonuclease activity, and these are then replaced by DNA. Question number 84. Which analytical tool was used by Hershey and Chase to demonstrate that DNA serves as hereditary material and not the protein? The options are 1, radio tracer technique. Number 2, X-ray diffraction analysis. Number 3, spectrometry. And number 4 is density gradient centrifugation. So here the correct option is the number 1, the radio tracer technique. They use the radioactive sulfur and the phosphorus in the experiment. Question number 85. The polymerase that synthesizes a polynucleotide chain in a template independent manner, note down, independent manner, is option 1, DNA polymerase 1. Option 2, DNA polymerase 3. The option 3 is RNA polymerase. Option 4, poly-A polymerase. So the correct answer is the first option, DNA polymerase 1. Question number 86. Discovery of ribozymes associated with number 1, RNA splicing. Number 2, transcriptional silencing. Number 3, translational silencing. Number 4, DNA ligase activity. The correct option is the number 1, RNA splicing. The RNA segments that cut themselves out of larger RNAs, so they are also called self-splicing introns. Question number 87. Identify the factor that terminates prokaryotic translation. The option number 1 is IF2 GTP. Sorry, it is 1, not I. 1F2 GTP. Option number 2 is 1F2. Option number 3 is RF1. Option number 4 is RG3. The correct answer is the option number 3, RF1. In prokaryotes, there are two factors, RF1 and the RF2. While in the eukaryotes, for the termination of the translation, there is a single factor called ERF1. Question number 88. In a charged tRNA, amino acid is linked at 3' and 5' and D loop or adjacent to the anticodon sequences. The correct option is the option number 1, 3' and. Question number 89. A human recombinant gene can be successfully translated in E. coli. This is because the option 1, genetic code is universal. Option 2, genetic code is degenerate type. Option 3, E. coli and human have similar translation factors. Option 4, E. coli and human have similar ribosomal organization. So here the correct one is the option number 3, E. coli and human have similar translational factors. Question number 90. Which one is used as genetic vector? The options are 1, lambda phage DNA, number 2, retroviral RNA, number 3, retroviral cDNA, and number 4 is RNA primer. The correct answer is the lambda phage DNA. Question number 91. In a P700 reaction center of chlorophyll, 700 denotes for 1, light wavelength, 2, number of reaction centers, 3, potential of the photosystem, 4, number of water molecule split. So it is the option 1, the light wavelength, the wavelength at which there is maximum absorption. Question number 92. During photosynthetic dark reaction, the inorganic carbon is fixed with option 1, ribulose 2,5 biphosphate. Second option, ribose 2,5 biphosphate. Option 3, ribulose 1,5 biphosphate. Option 4, ribose 1,5 biphosphate. So here the correct one is the option number 3, which is the ribulose 1,5 biphosphate. It is also commonly called rubisco. Moving to the next question, the question number 93. Identify the CU stands for copper, containing photosynthetic pigment. The option 1, chlorophyll. Option 2, plastocyan. Option 3, thioredoxin. Option 4, ferredoxin. Here the correct one is the option number 2, which is the plastocyan. The plastocyanin is a metalloprotein. It contains copper. Question number 94. In eukaryotes, the first transcript synthesized by RNA polymerase II is referred as option 1, messenger RNA, option 2, snRNA, option 3, hnRNA, option 4 is the tRNA. So here the correct is the second one, it is the snRNA. Question number 95. The conformation of 2' deoxyribose in a DNA double strand is 1. Chair type, 2. boat type, 3. fluoronosrin type, 4. pucker type. The correct one is the 4. puckered type. Question number 97. Maximum number of electrons in a subshell with L is equal to 3 and N is equal to 4 is number 1 10, number 2 12, 14 or 16. The correct answer is the 14. This L is equal to 3, N is equal to 4. It represents the 4f orbital and maximum number of the electrons can be 14. Question number 98. Magnesium 2 plus Mg2 plus is isoelectronic with Ca2 plus, Na plus, Zn2 plus, Cu2 plus. Here the correct answer is the option number 2, which is the Na plus. Isoelectronic means having the same number of the electrons. Question number 99. How many stereoisomers of the 3-bromo-2-butanol exist? The options are 2, 4, 3 and 1. The correct option is the option number 2, which is the 4. There can be the 4 stereoisomers of 3-bromo-2-butanol. Question number 100. The isomers which can be interconverted through rotation around a single bond are number 1 conformers, number 2 diastereomers, number 3 enantiomers, number 4 positional isomers. So the correct answer is the number 1, which is the conformers. 101. Standard enthalpy change of combustion occurs when 1 mole of a substance is burnt in excess of option 1 nitrogen, option 2 oxygen, option 3 carbon dioxide, option 4 is helium. The correct one is the option number 2, which is the oxygen. Question number 102. CH3CHO and C6H5CH2 CHO can be distinguished chemically by, these are the aldehydes. Number 1 Tollens reagent test, number 2 Fehling solution test, number 3 Benedict test, number 4 is the iodoform test. The correct answer is the number 4. This is the iodoform test. Iodoform reagent gives yellow crystalline precipitate with acetaldehyde. Question number 103. The enzyme tyrosinase is activated by the option 1 iron, option 2 copper, option 3 zinc and option 4 is potassium. The correct answer is the option number 2. It is the copper. Question number 104. In hemoglobin, the transition from T state to R state is triggered by Fe2 plus binding, heme binding, oxygen binding, subunit association. The correct answer number 3, oxygen binding. In hemoglobin, there is transition from the T state to the R state, which is triggered by binding with the oxygen. Question number 105. Ethylene glycol reacts with dimethyl terephthalate to form the number 1 nylon 66, number 2 Teflon, number 3 Orlon, number 4 is Dacron. The correct one is the number 4 Dacron. Dacron is a polyester formed by condensation polymerization of a diol and a diacid. Question number 106. The number of asymmetric carbon atoms in the alpha D glucopyranose molecule is. The options are 2, 3, 4 or 5. Here the correct answer is the option number 4, which is the 5. As the number of chiral carbon atoms are 5. Question number 107. Which is a disaccharide? Number 1 glucose, number 2 maltose, number 3 fructose, and the option number 4 is cellulose. So the disaccharide is maltose. The monomers of maltose are glucose. Question number 108. Which of the hydrogen A to D in the following molecules gives a triplet signal in a normal H NMR spectrum? So here this structural formula is shown, and the carbons are A, B, C, D, and the options are hydrogen A, hydrogen B, hydrogen C, and the hydrogen D. The correct answer is the option number 3, which is the hydrogen C. The signal which results from this spin-spin coupling with two equivalent adjacent hydrogen atoms. Question number 109. Which one of the following set of quantum numbers represent highest energy? Number 1, n is equal to 2, l is equal to 1. The option 2, n is equal to 3, l is equal to 2. Option 3, n is equal to 3, l is equal to 1. The option 4, n is equal to 2, l is equal to 0. So here the correct one is the option number 2, where n is equal to 3 and l is equal to 2. The set of quantum numbers is determined as we add the n and n. So n plus l, the value of the n plus l is maximum in the option number 2. So it will be having the highest energy. Question number 110. The strength of hydrogen bond is intermediate between option 1 van der Waal and covalent, option 2 ionic and covalent, option 3 ionic and metallic, option 4 is metallic and covalent. The correct answer is the option number 1 van der Waal and the covalent. Question number 111. For a reaction to be spontaneous, the following is essential to be negative. The option 1 is delta H minus T delta S. The option 2 delta H plus T delta S. Option 3 is delta H. Option 4 is delta S. So here the correct answer is the option number 1. For a spontaneous reaction, the free energy should be negative, and the free energy change is represented by delta H minus T delta S. Question number 112. When ice melts into water, entropy becomes 0, decreases, increases, or remains same? The answer is the option number 3, which is increases. Question number 113. The value of free energy change at equilibrium is number 1 positive, number 2 negative, number 3 0, and number 4 not definite. The correct answer is the option number 3. It is 0, because when the reaction is at equilibrium, there is no more work which can be done. So free energy is 0. Moving to the next question, which is a question number 114. Which one of the following is not a hard base? Number 1 ammonia NH3, number 2 water H2, number 3 is chloride ion, number 4 is the cyanide ion, not a hard base. So the correct one is the option number 4, which is the cyanide ion. So the cyanide ion is a hard base is a Lewis base with a weakly polarizable electron donor center. Question number 115. Hg2 plus, Hg is for mercury, Hg2 plus is classified as 1 soft acid, 2 hard acid, 3 soft base, 4 is the hard base. So it is a soft acid. The correct answer is the 1 soft acid. Soft acids, they have a low charge density and low ionic charge and a greater ionic size. Question number 116. Winkler method is used to determine number 1 dissolved oxygen DO, number 2 biochemical oxygen demand BOD, number 3 organic carbon OC, number 4 elemental carbon EC. The correct answer is the option number 1. It is the dissolved oxygen. The question number 117. The smog is generally caused by the presence of number 1 oxygen and ozone, O2 and O3, number 2 NOx and SOx. Actually it should not be X, it should be 2. Number 3 O2 and N2, number 4 O3 and N2. The correct option is the second option, NO2 and SO2. The smog is air pollution and there are the volatile organic compounds like the sulfur dioxide and nitrogen dioxide which are released in the air and they remain suspended as the fine particles. Number 118. The prefixes Z and E stand for number 1 Ziegler-Ehrhardt, number 2 Zitter-Erythro, number 3 Zirco-estrogen, number 4 is Zuckerman antigen. The correct answer is the number 4, which is the Zuckerman antigen. It is used in the case of the alkenes. If there are two priority groups which are on the opposite side, it is called the E, the antigen, and if it is on the same two sides, then it is called the Zuckerman. Question number 119. Bakelite is a cross-linked polymer of 1 phenol, 2 formaldehyde, 3 both phenol and formaldehyde, and 4 is the wool. So the correct answer is the option number 3, both phenol and formaldehyde. It is a thermosetting polymer. The question number 120. Alpha D glucose and beta D glucose are number 1 anomeric sugar, number 2 apimeric sugar, number 3 these are the position isomers, number 4 these are the functional isomers. The correct answer is the number 1. These are the anomeric forms. It should be anomers, not the anomeric sugar. These are the anomers. The alpha D glucose and beta D glucose, they are actually stereoisomers that differ in the three-dimensional configuration of the atoms and order groups at one or more position. So these are the anomers. So these were the 60 questions which were asked in the BHU entrance examination in 2018. Hope this will help you. Stay tuned. Best wishes. Bye. Take care.